Hello everyone, welcome to VLearn and welcome back to the class Introduction to the Genetics. In the last class of genetics video, we studied about the principle of inheritances and we studied about principle of segregation and monohybrid cross. In today's class, we are basically going to study about the different types of cross. The first type of cross we are going to study about in this class is back cross. So if we talk about back cross, let us say a homozygous stole plant is crossed with homozygous recessive plant. This homozygous stole and homozygous recessive plant, as you know, the gamete of the homozygous stole would be capital T and all the gamete of this homozygous recessive plant would be small t. In the F1 generation, when these gametes mixes up, these ones produces a generation which is heterozygous. All the plant in this generation are, of course, heterozygous tall. So all the plants are basically heterozygous tall. If this heterozygous tall plant is crossed with either one of the generation, either one of the parental generation, then it is called back cross. Then it is said to be back cross. If we cross this homozygous tall plant, heterozygous tall plant, if we cross this heterozygous tall plant with homozygous dominant plant, then it is said to be outcross. But if we cross this heterozygous tall plant with homozygous recessive plant, that is recessive parent plant, then it is said to be test cross. So basically you can say there are two types of back cross. Both the cross are basically the back cross, the out cross and the test cross. Both of these cross are basically the part of back cross. But if we cross the F1 generation with homozygous tall plant, then it is said to be outcross. But if we cross heterozygous tall plant, then it is test cross. The application part of this back cross. So if we know that if we cross F1 generation, that is a tall plant, heterozygous tall plant, capital T, small t, with another plant, that is small t, then this type of cross is basically what we called test cross. This is test cross. Now the application part of the test cross. Application part. What is the application? Let us say there is a tall plant, capital T and small t. This particular plant is basically heterozygous tall. This particular plant is basically heterozygous tall. The other character of a tall plant could be, the other genotype of the tall plant could be, one genotype could be heterozygous tall. The other genotype of this plant could be homozygous tall, capital T, capital T. So capital T, heterozygous tall plant, homozygous tall plant, sorry. So this plant is homozygous tall. But how would we know that this particular tall plant is either heterozygous tall or homozygous tall? We can basically know this by test cross or back cross. So basically this test cross is particularly used for knowing the genotype knowing the genotype of a tall plant, or you can say the dominant organism. So this test cross is basically used for knowing the genotype of dominant organism. Genotype. So how would we know the genotype of an organism? Let us say, for knowing the genotype, we have to do the cross. A tall plant is crossed with a dwarf plant. The genotype of dwarf plant would be small t and small t, but the genotype of the tall plant could be capital T, or it could be capital T small t. By this cross, we can particularly know the genotype of this tall plant. Genotype of this particular tall plant. So let us say we have to know the genotype of this plant. What is the genotype of this plant? So upon cross, the genotype of this plant could be the genotype of this tall plant could be homozygous dominant. This plant could be homozygous dominant. The genotype of this plant could be heterozygous dominant. What we do is we have to check this tall plant, with the test cross. So we cross, if the genotype is homozygous dominant, we cross this plant with the homozygous dwarf, that is small t small t. We don't know the genotype of this plant. Let us say this genotype is homozygous dominant. If it is homozygous dominant, all the plant in the next generation would be tall plant. All the plants would be tall plants. 100% of the plant would be tall plants. No plant, not a single plant would be there having the genotype small t, or the dwarf nature or the dwarf phenotype. So all the plants, that is 100% of the plant, if all the plants are dominant, if all the plants are dominant plant, then it is said to be homozygous dominant. But if all the plants are not dominant, then it is said to be heterozygous dominant. Let us say the genotype is capital T small t of this particular